Welcome to part 3 of 3 looking at chapter 7 on cellular respiration. The first two parts of this chapter we spent time looking at cellular respiration when oxygen is present. On this third part we're going to be looking at the second part of this chapter, looking to see what happens when oxygen is absent from the environment or there's a lack of oxygen. And there's two things that can happen. You're either going to have anaerobic respiration, or you can also have something called fermentation that occurs. So in anaerobic metabolism, these are for environments that lack oxygen or during oxygen deficits. So in environments that lack oxygen, there's a lot of anaerobic environments. Bacteria are a really good example of organisms that live in anaerobic environments. In fact, some bacteria are actually killed by oxygen, so they have to live in an anaerobic environment. So for example, here we have uranium waste bacteria, so bacteria that break down uranium. They are killed by oxygen, so they do this alternative form of cellular respiration. In oxygen deficits, example here we have a cheetah running, or if you've ever worked out or gone running, you get that really heavy breathing, kind of lack of oxygen feeling during workouts. That's when you have an oxygen deficit in your muscles. So your muscles are going to start to convert into an anaerobic metabolism to keep making ATP so that you can keep on working out. So in order to address these anaerobic conditions, either when you just don't have oxygen or you have a lack of oxygen, there's two things that can occur. So one strategy is that instead of using oxygen at the very end of the electron transport chain as the final electron acceptor, you can use some other substance other than oxygen. So you remember, that's that point where I pointed out, here's our oxygen, this is where we're using it. Some organisms, they use something else to catch that final electron. Or the other strategy is to only produce ATP using substrate level phosphorylation, or basically only doing glycolysis, and not going into breakdown of pyruvate, citric acid cycle, and oxidative phosphorylation. So other acceptors using this first strategy: Ecoli, which is a species of bacteria found in the intestinal tract of mammals. Ecoli, instead of using oxygen as a final electron acceptor, if it's in an anaerobic environment, it's going to use a nitrate molecule to catch that final electron. So hopefully this image looks familiar. We talked about it in part 2 of this chapter. So this is again that fourth step where we take our NADHs and our FADH2s, we extract electrons out of them and hydrogen ions. You set up your hydrogen ion gradient to make your ATP using that chemiosmosis. Your electron moves through the electron transport chain to help set up that gradient. That electron has to be caught. Instead of using oxygen, it's caught using a nitrate molecule in this example. So this is a really good strategy because you can still make up to 38 ATP using that chemiosmosis even when you don't have oxygen available. So very efficient strategy. The other strategy is going to be the fermentation. So many organisms, they have to use oxygen as that final electron acceptor. There's no other choice. They can't use any other type of molecule. So when there's no oxygen available, all these cells can do is glycolysis. So they can only make ATP using glycolysis. And if you remember when we talked about glycolysis, you get 2 ATPs created by substrate level phosphorylation. So it's not as efficient, but we're still creating ATP, so the cells can still be living. They're still making energy using this fermentation method. The problem we run into is that we also create 2 molecules of NADH during glycolysis, which are not going to be used at all. So we need a way to take those hydrogens back off to regenerate our NAD+, so that we can make more NADHs. In order to get rid of our NADH molecules, muscle cells in humans, we produce lactic acid or lactate. This is the acid that makes your muscles really sore, and it takes a while for your muscles to kind of clean up all the lactic acid when you're done working out. Other organisms like yeast, for example, which is in the fungi kingdom, instead of making lactic acid, they make ethanol, which is drinking alcohol that you find in wine and beer for that. So here, glycolysis, again, if you're working out, we're going to produce lactic acid, which is going to make you sore. In other organisms, so yeast and some bacteria, they're going to produce ethyl alcohol or ethanol, which is in the wine or the beer. Okay, so that's what's going to happen when oxygen is absent. You're either going to have that first strategy, so using something other than oxygen as your final electron acceptor, or you can go into the fermentation process. And fermentation, you're going to end up, the cells are going to have to produce some sort of acid or alcohol in order to kind of take care of all the extra NADH that was created during glycolysis. That's not going to be used in the oxidative phosphorylation, because that's not going to happen in those organisms that do the fermentation process. So, and just to leave off, I just want to remind you about the slide because it is really, really important. This summarizes kind of what is occurring in this chapter. Remember, really focus on inputs for each of the 4 steps, the output, so what's coming out of them, where those steps are actually occurring in the cell, and then our ATP coming out the bottom, the 2 ways that you can have ATP synthesis.